Hello students, today's topic is recombinant DNA technology. This technology is used for genetic engineering. There are many examples where recombinant DNA technology has been used. For example, a bacteria which is engineered, E. coli bacteria is there, to produce human insulin hormone which fights for diabetes. Yeast cells were made to produce hepatitis B vaccine. Then in case of BT cotton, so in this cotton plant, it was made insect resistant to Bacillus thuringiensis. Then engineered bacteria to clean up environmental waste such as polythene. Now, what are the various steps which are required for the formation of recombinant DNA? So, before this, let me tell you that first recombinant DNA molecules to be generated using the above mentioned procedures were established by combined efforts in 1973 by the molecular biologists Paul Berg, Herbert Boyer, Annie Chang, and Stanley Cohn. By the methodology of recombinant DNA technology, we can produce large amount of DNA, and that process is known as gene cloning. We can also produce large amount of protein expressed by the insert, because we know that this DNA, it transcribes to form RNA and after translation forms protein. So we can get large amount of DNA as well as protein also. Now the first step is that we have to isolate DNA fragment containing a gene of interest that needs to be cloned called the insert. So we'll isolate desired DNA and digest that with restriction enzyme. Then generation of recombinant DNA molecule by insertion of the DNA fragment in a carrier DNA molecule called vector. Vector is also digested with same restriction enzyme. We can use different types of vectors like plasmids, phages, cosmids, BAC, YAC, plant and animal viral vectors, which can replicate themselves within a host cell after the digestion with same restriction enzyme, so that we can generate their complementary sequences. Then both are ligated with the help of DNA ligase, and finally transfer of our DNA into an E. coli host cell by the process of transformation. Here selection of only those host cells carrying the rDNA and allowing them to multiply. Now it will be clear from this figure that this is the desired fragment. This is the vector DNA like plasmid. When it is digested with same restriction enzyme, then it will open and this fragment can be inserted inside it with the help of DNA ligase enzyme, and this structure is known as recombinant DNA, hybrid DNA or chimeric DNA. After this recombinant DNA is transformed in host cell, bacterial cell, and bacterial cell is having its own DNA along with this rDNA. This rDNA will replicate inside the bacterial cell, and in this way we can get large number of this rDNA from host cell. Then what are the tools of recombinant DNA technology? Restriction enzymes, other enzymes like alkaline phosphatase and DNA ligase, vectors and host cells. These are the four major things which are required for the formation of recombinant DNA. First, we'll deal with restriction enzymes. These enzymes are also known as molecular scissors. Why? Because they are used to produce cuts at the molecular level. Now, these enzymes, they naturally present in bacteria as the defense system and also called as restriction modification system. Now, how we'll tell you that how these enzymes are present in bacteria and how they provide defense to them. Now, restriction enzyme part of this system restricts the entry of phage molecules by digesting them in bacteriophage. So, whenever any virus comes to infect any bacteria, and what happens? Virus coat protein remains outside the cell. Only DNA or RNA moves inside, and this restriction enzyme of restriction modification system identifies the foreign DNA, it digests the foreign DNA and inactivates that. Then modification enzyme adds methyl group to one or few bases of sequences which are recognized by restriction enzyme to avoid cutting off their own sequences. So this is how host DNA, because restriction enzymes, they must have that recognition sequences inside the host DNA also. Now to prevent our own DNA, bacterial own DNA from digestion, methyl group is added. Different bacteria of their own, they are having their own restriction endonucleases and corresponding methylase enzymes. Restriction enzymes were first discovered and studied by the molecular biologist Arbor, Smith, and Nathans, for which they were awarded the Nobel Prize in 1978. Today, more than 1,000 restriction enzymes are available for genetic engineering purposes. Then there are three types of restriction endonucleases: type 1, type 2, and type 3. Now, type 1 and type 3, like other restriction enzymes, they recognize specific sequences but they produce cuts somewhere else. So, these are not specific in nature. So, both of them cannot be used in recombinant DNA technology. Now, the third type is type 2. These enzymes recognize specific sequence which is 4 to 8 base pair long and produces cut within the same sequence. Now, such enzymes recognize palindromic sequences. Palindromic sequences means sequences are same but they are in opposite direction, which means that the sequence in both DNA strands at this site read same 5' to 3' direction. Now, this is about the nomenclature. There is one restriction enzyme which is commonly present inside the E. coli, that is EcoR1. So you can check from this thing that the first alphabet is capital E. It is represented E. coli, that is genus, and it should be in caps. Then CO is representing species, coli. So here CO, second and third alphabet is taken from the species. Then fourth alphabet, which is again capital, is name of strain, RY13. Then I is the Roman one, and the number designated with Roman numeral indicated that it was the first enzyme to be isolated from this particular strain of bacteria E. coli. Now, if we check, now we have studied about types of restriction enzymes. Now, there are restriction enzymes which can produce different types of cuts. Now, if we check we know that restriction enzymes, they can recognize a sequence which is 4 to 8 base pair, and they have to produce cuts in the palindromic sequences. If we check in the middle of each and every palindromic sequence, there is line of symmetry. Now, if restriction enzymes, they produce cut along the line of symmetry, then we get flush or blunt ends. Similarly, if these restriction enzymes, they produce cut away from line of symmetry at the ends, then these produce sticky or cohesive ends. So these are various examples of restriction enzymes which can either produce blunt ends if they are cutting along the line of symmetry, or if they are cutting away from the line of symmetry, then they produce sticky or cohesive ends. Next is application of restriction enzyme, that is RFLP, restriction fragment length polymorphism. Now, DNA isolated from an individual organism, they have unique sequence. Even the members within a species, they differ in some parts of their sequence. Restriction sites would also vary, and hence if DNA from a given individual was subjected to digestion with a restriction enzyme, the fragments generated would vary when compared with another individual's DNA similarly digested. Now, this variation in size or length of the restriction enzyme generated fragments among individuals within a given species is termed as RFLP. What is the major application of RFLP? That is DNA fingerprinting analysis. Individuals except identical twins, they vary in their RFLP pattern as indicated schematically in agarose gel electrophoresis. Hence, the term DNA fingerprinting is used, and this is the basis of major technique used in forensic science to identify and relate individuals. Now, it will be clear from this figure. Like this is individual A, this is individual B. isolate their DNA and digest them with restriction enzyme. You will see that this restriction enzyme is same, but in this individual, it is producing these fragments of different sizes, and because they can cut at different sites. Similarly, here different size and different number fragments are being produced. So in this way, we can know about the DNA fingerprints of individual, and this process can be used in forensic science also. Thank you so much, and if you like my video, do subscribe and like, and you can share your comments also. Thank you.